All right, ready to dive into this mystery of mysteries?
Yeah.
Speciation.
So cool.
Whole chapter on how new species arise.
Yeah.
And you've given us some pretty great excerpts and diagrams from a biology textbook.
Yeah.
I'm excited to see what we uncover.
It's amazing how much we've learned, like, since Darwin first pondered this question, you know?
Right.
It's like piecing together a really intricate puzzle. We're starting to get the full picture of how life diversifies.
And you've provided us with some really cool puzzle pieces.
Yeah.
From flightless cormorants on the Galapagos Islands to this big bird finch that's emerged through hybridization.
Right?
It's like evolution is constantly coming up with new and surprising ways to, like, create diversity.
Exactly. And one of the key pieces of this puzzle is understanding how we even define a species.
Right.
There are a few ways to do this,
Yeah.
but the biological species concept is a good place to start.
Right. The idea that a species is a group of populations that can interbreed
Yeah.
and produce viable, fertile offspring. It's
Right.
like a club with exclusive membership. Only those who can successfully reproduce are allowed in.
A great analogy. But like any exclusive club, there's always a few exceptions and nuances, you know?
Oh, of course. Yeah.
For example, this concept doesn't work so well for asexual organisms
Right.
or fossils.
Yeah.
We can't exactly observe dinosaurs mating
Right.
to see if they were reproductively isolated.
True. So there are other ways to define a species, right?
Right,
Like
yeah.
focusing on physical characteristics
Right.
or their roles in the ecosystem.
Exactly. The morphological species concept relies on observable physical traits.
Okay.
Whereas the ecological species concept looks at a species' niche, its role in the ecosystem.
Right.
These concepts are useful when the biological species concept isn't a good fit,
Okay.
like when dealing with fossils or asexual organisms.
Right. So different definitions for different situations. That all depends on what we're trying to understand. But for our deep dive into speciation, the biological species concept will be our main guide, with its focus on reproductive isolation.
Precisely. Now the real magic happens when we look at the mechanisms that drive this reproductive isolation.
Okay.
Think of it like barriers that prevent gene flow between populations.
Okay, so these barriers are what keep those species lineages on separate evolutionary paths.
Exactly.
What kinds are there?
We can categorize them as prezygotic or postzygotic barriers,
Okay.
depending on whether they act before or after the formation of a zygote.
Prezygotic barriers. Those are the ones that prevent mating or fertilization from happening in the first place.
Exactly. And there are some really fascinating examples.
Yeah.
Think about the blue-footed boobies of the Galapagos Islands.
Yeah.
Their elaborate mating dance, where males lift their bright blue feet,
Right.
acts as a prezygotic barrier.
Okay.
Females won't even consider mating with a male who doesn't perform this, like, species-specific ritual.
So even a silly dance can be a crucial part of keeping species separate.
It's pretty amazing.
What other prezygotic barriers are out there?
Well, there's habitat isolation, where two species might live in the same geographic area, but occupy different habitats, preventing them from encountering each other.
Right.
The apple maggot fly is a great example. One group feeds and mates on hawthorn fruits, while another prefers apples.
Oh, interesting.
They're in the same area, but their different preferences keep them reproductively isolated.
So even a change in diet can set the stage for speciation.
Absolutely.
What about those skunks
Yeah,
that mate in different seasons? Is
Yeah.
that another type of prezygotic barrier?
absolutely. That's temporal isolation.
Okay.
Think about the western spotted skunk and the eastern spotted skunk.
Okay.
Their breeding seasons don't overlap.
Oh, wow.
So even though their ranges overlap, they're unlikely to interbreed. It's all about timing.
Fascinating. And then there's the textbook example of mechanical isolation.
Right.
Those snails whose shell spirals determine the orientation of their reproductive parts. Talk about incompatible puzzle pieces.
It's a striking example of how physical incompatibilities can prevent mating altogether.
Right.
And let's not forget gametic isolation,
Right.
where the sperm and egg of different species might be incompatible, preventing fertilization even if mating occurs.
Right.
It's like having a lock and key that just don't match.
So we've got this whole arsenal of prezygotic barriers.
Yeah.
From elaborate dances to incompatible reproductive parts,
Right.
all working to prevent different species from mixing their genes.
Right.
But what about after fertilization? That's where the postzygotic barriers come
You
in.
got it. These barriers act after a hybrid zygote has formed,
Okay.
often resulting in reduced viability or fertility of the hybrid offspring.
Like the classic example of the mule.
Yes.
A hybrid between a horse and a donkey.
Yeah.
Strong and hardy, but usually sterile.
A perfect example.
Yeah.
Reduced hybrid fertility is a major postzygotic barrier.
Right.
And let's not forget hybrid breakdown, where the first generation of hybrids might be fertile.
But their offspring experience reduced fertility or viability.
Oh, wow.
It's like a delayed form of reproductive isolation.
So even if hybrids are initially successful, their lineage might fizzle out over time
Right.
due to these postzygotic barriers.
Right.
It's all about maintaining the integrity of those species boundaries.
Absolutely. Now, understanding these barriers is crucial for understanding how new species arise in the first place.
Right.
And this can happen in two main ways: allopatric and sympatric speciation.
Okay, I'm ready to delve into the hows of speciation.
Right.
What's the difference between these two pathways?
Think of allopatric speciation as speciation with a physical barrier.
Okay.
Imagine a mountain range rising, a river changing course, or a group of individuals colonizing a new island.
Right.
These events lead to geographic isolation,
Okay.
separating a population into two or more subgroups that can no longer interbreed.
Okay, so it's like long-distance relationships for species.
Yeah. Distance makes the heart grow fonder, but it can also lead to evolutionary divergence.
A great way to put it. Over time, these isolated populations experience different environments, different mutations, and different selective pressures.
Yeah.
This leads to genetic divergence.
Right.
And eventually, they may become so different that they can't interbreed even if they were to come back into contact.
That's amazing. So physical separation is the key driver in allopatric speciation.
Yes.
But what about sympatric speciation?
Yeah.
It's trickier, right?
Yes.
Because it happens without geographic isolation.
Exactly. Sympatric speciation is like trying to separate two groups of people who live in the same house.
Right.
It requires some creative evolutionary maneuvering.
All right, I'm intrigued. How does sympatric speciation even work? What are some of those creative evolutionary maneuvers?
One of the most dramatic mechanisms is polyploidy, where accidents during cell division can result in extra sets of chromosomes. Imagine a plant suddenly doubling its chromosome number. It might be able to self-fertilize or mate with other polyploids,
Right.
but it can't successfully reproduce with the original plants.
Oh, wow.
Instant reproductive isolation.
Whoa. That's like a genetic shortcut to speciation.
Yeah!
Does this happen often in nature?
It's more common in plants than animals,
Okay.
but it's played a huge role in plant evolution.
Wow.
Many of the crops we rely on, like bread wheat, are actually polyploids.
Really?
It's a testament to the power of polyploidy to drive speciation
Wow.
and even influence our own food supply.
I never thought about it that way.
Yeah,
We're literally eating the results of polyploidy.
we are.
Are there other ways sympatric speciation can occur?
There are. Sexual selection can play a role too.
Okay.
Think about those cichlid fish in Lake Victoria.
Yeah.
It was once home to hundreds of cichlid species.
Hundreds of cichlid species in just one lake.
Yeah.
That's incredible. How did that happen?
Scientists believe that sexual selection played a major role. Imagine female cichlids choosing mates based on their coloration. Over time, a preference for certain colors could lead to the formation of distinct groups, eventually becoming separate species. It's like fashion driving evolution.
I love that idea. So it's not just about physical barriers or dramatic genetic changes. Even something as simple as a color preference can lead to new species.
Exactly. And then there's habitat differentiation, where a subpopulation within a species starts exploiting a new habitat or resource. Remember those apple maggot flies?
Yeah.
Their shift from hawthorns to apples is a prime example of this.
Right. That change in diet created a new habitat, setting the stage for the apple-feeding flies to evolve different traits and behaviors, further separating them from their hawthorn-feeding ancestors. It's amazing how a simple preference for a different type of fruit can kick off the process of speciation.
It really highlights the intricate interplay between ecology and evolution. Now, all of these mechanisms, allopatric speciation, sympatric speciation, polyploidy, sexual selection, habitat differentiation, they all contribute to the incredible diversity of life we see today.
It's like this grand evolutionary tapestry woven over millions of years, with each thread representing a unique lineage that's connected to all the others.
A beautiful analogy. And to make this tapestry even richer, we have to consider what happens when those threads intertwine. That's where hybrid zones come in.
Hybrid zones, those are areas where different species with incomplete reproductive barriers meet and interbreed, right? It's like the evolutionary equivalent of a melting pot.
Precisely. Hybrid zones are fascinating natural laboratories where we can study the forces that drive speciation. They can take many forms, sometimes appearing as narrow bands where two distinct habitats meet. A great example is the hybrid zone between the yellow-bellied toad and the fire-bellied toad in Europe.
So it's like a geographic border between two species with some adventurous individuals crossing over and interbreeding. What happens to those hybrids? Do they thrive or do they struggle?
Well, hybrids often face challenges. They might have reduced survival rates or be less fertile compared to their parent species. It's like mixing two recipes together. Sometimes you end up with a culinary masterpiece, and sometimes not so much.
That's a perfect way to put it. So what happens when those hybrid offspring are less fit? Does natural selection play a role?
Absolutely. Natural selection favors individuals who are less likely to produce those less fit hybrids. This can lead to the reinforcement of reproductive barriers, making it even harder for the two species to interbreed. It's like evolution is trying to maintain the integrity of those species' lineages.
So hybrid zones can actually contribute to the process of speciation, even though they involve interbreeding between different species. That's really counterintuitive. But I imagine these zones don't always stay the same. What happens when environmental conditions change?
That's a great point. Environmental changes can shift those hybrid zones or even create new ones. Take climate change, for example. As temperatures rise, some species are expanding their ranges northward. This can lead to the formation of novel hybrid zones where species that were previously geographically separated now come into contact and interbreed.
That's like climate change is shuffling the evolutionary deck, leading to new interactions and potentially new hybrid zones. And didn't you mention that hybridization can sometimes be beneficial? That seems paradoxical given that we've been talking about how hybrids are often less fit.
It is a bit of a paradox, but it highlights the complexity of evolution. Sometimes hybridization can introduce beneficial genes from one species into another. There's evidence suggesting that hybridization between certain mosquito species might be contributing to the spread of insecticide resistance.
So while hybridization can sometimes lead to evolutionary dead ends, it can also be a source of new genetic variation that allows species to adapt to changing environments. It's a reminder that evolution isn't always predictable or straightforward.
Exactly. Now let's shift gears and consider the tempo of speciation. How quickly can this process happen? Does it always take millions of years?
That's a great question. I remember learning about gradualism versus punctuated equilibrium in evolution. Do those concepts apply to speciation as well?
Absolutely. The fossil record reveals evidence for both gradual change over time and periods of apparent stasis punctuated by sudden bursts of evolutionary change. This tells us that speciation can occur at different speeds depending on the circumstances.
So it's not a one-size-fits-all process. Some species might take their time evolving gradually over long periods, while others might experience these rapid bursts of speciation, almost like evolutionary sprints. What are some examples of these different tempos?
Well, the classic example of rapid speciation is the wild sunflower Helianthus anomalus. It arose from the hybridization of two other sunflower species in a relatively short period of time.
So hybridization can actually lead to the formation of a brand new species. That seems counterintuitive, given that we often think of hybridization as leading to less fit offspring.
It is remarkable. In the case of Helianthus anomalus, the hybrid offspring were initially less fertile, but after a few generations, their fertility increased dramatically. Scientists think that natural selection acted to eliminate incompatible gene combinations from the hybrid genome, essentially fine-tuning it for success. This led to a new species that's both ecologically distinct and reproductively isolated from its parent species.
It's amazing how hybridization can sometimes be a creative force in evolution, leading to the birth of new species. But how long did this process take?
It happened surprisingly quickly, within a few thousand years. And to make things even more interesting, scientists have even recreated this process in the lab, providing further evidence for the role of hybridization in rapid speciation.
It's incredible to think that we can witness these evolutionary processes unfolding in real time, both in nature and in the lab. It really speaks to the power of science to illuminate the mysteries of life. But what about those cases of slower speciation? Do we have any examples of that?
The fossil record provides plenty of examples of gradual change over millions of years where species slowly diverge from their ancestors. One well-studied example is the evolution of horses. Over millions of years, horse lineages gradually transitioned from small multi-toed browsers to the large single-toed grazers we see today.
So we have this spectrum of speciation tempos, from rapid bursts driven by hybridization to slow and steady changes over vast stretches of time. It seems like evolution has a toolbox full of different strategies for creating diversity.
That's a great way to think about it. Now let's delve into the genetic underpinnings of this diversity. How many genes need to change for a new species to arise? Is it just one or two, or could it be hundreds?
That's a fascinating question. I remember reading about how a single gene can sometimes have a major impact on reproductive isolation, like those Japanese snails with the spiraling shells.
Exactly. A single gene mutation can sometimes be enough to create a significant reproductive barrier. But in other cases, speciation might involve changes in many genes, making it much more difficult to pinpoint the specific genetic changes responsible for reproductive isolation.
So it's not always a simple one-to-one relationship between genes and speciation. It's more like a complex interplay of multiple factors, with the number of genes involved varying depending on the species and the specific mechanisms driving their divergence.
Precisely. It's a reminder that evolution is a master tinkerer, working with whatever genetic material is available to create new and diverse forms of life. And as we delve deeper into the genetics of speciation, we're starting to uncover the intricate molecular mechanisms that underlie this incredible process.
I'm eager to learn more about these molecular mechanisms. What are some of the latest findings in this area?
One exciting area of research is focused on identifying the specific genes that contribute to reproductive isolation. Scientists are using a variety of techniques, from comparing the genomes of closely related species to conducting experiments that manipulate gene expression to pinpoint the genetic changes that drive speciation.
So it's like we're zooming in on the evolutionary process, going from observing patterns at the species level to understanding the molecular details that make it all possible. What are some examples of genes that have been linked to speciation?
Well, one fascinating example involves a gene called agouti, which influences coat color in mice. Researchers have found that differences in the agouti gene between two closely related mouse species contribute to their reproductive isolation. Mice with different coat colors are less likely to mate with each other, demonstrating how even seemingly subtle genetic changes can have a significant impact on reproductive behavior and ultimately speciation.
It's like a tiny genetic tweak that can ripple through the entire system, influencing everything from appearance to mating preferences.
Exactly. And as we continue to unravel these genetic intricacies, we're gaining a deeper understanding of the mystery of mysteries, how new species arise and contribute to the breathtaking diversity of life on Earth.
It's incredible to think that tiny genetic tweaks can have such profound effects on the evolution of new species. But you mentioned earlier that sometimes it's not just one gene, but an accumulation of smaller genetic differences that leads to speciation. Can you elaborate on that?
What's fascinating is that in some cases, it's the interaction of multiple genes that creates the barrier to reproduction. We see this in the hybrid sterility of certain fruit flies. It's not a single gene that causes the problem. It's a complex interplay of several genes.
So it's like a delicate balance. Multiple genetic elements have to be just right for successful reproduction, and if that balance is disrupted, speciation can occur.
Exactly. And that complexity makes it even more challenging to pinpoint exactly which genetic changes are responsible for reproductive isolation. It's like trying to solve a puzzle with multiple missing pieces.
It sounds like the genetics of speciation can be incredibly intricate. But this just makes the study of evolution even more captivating.
It certainly does. And remember, speciation isn't just a historical phenomenon, it's an ongoing process. New species are constantly emerging while others fade into extinction. It's a dynamic dance of life that's been happening for billions of years.
Thinking about it that way makes me wonder about the implications for conservation. If we're losing species faster than new ones are arising, that's a serious issue.
You've hit on a crucial point. Understanding speciation gives us insights into how species adapt and evolve, which is essential for effective conservation efforts. It helps us identify those species that might be more vulnerable to extinction and develop strategies to protect them.
It underscores the interconnectedness of all life on Earth. Speciation is the process that has given rise to the incredible biodiversity we see today, and understanding it is crucial for preserving this natural heritage for future generations.
Well said, and as we've explored in this deep dive, speciation is a multifaceted process influenced by geographic isolation, genetic changes, and a variety of ecological factors, from the elaborate mating rituals of blue-footed boobies to the subtle genetic tweaks that influence coat color in mice. The mechanisms of speciation are as diverse as the species themselves.
This deep dive has truly been a journey of discovery, revealing the intricate ways in which life diversifies and adapts. We've uncovered the mystery of mysteries, exploring how new species arise, the barriers that keep them separate, and the genetic underpinnings of this extraordinary process.
And perhaps the most intriguing takeaway is that we've only just scratched the surface. There's still so much more to learn about the mechanisms of speciation, the interplay of genes and environment, and the implications for conservation in a rapidly changing world.
Indeed, the story of speciation is still being written, and it's a story that we're all a part of as we continue to explore this fascinating field. We're sure to uncover even more remarkable insights into the evolution of life on Earth.
So until next time, keep those minds curious.